the chromatography consumables group. I am the account manager for Florida, Mississippi, and Alabama. Chromatography consumables go from anywhere between auto sampler vials, columns for your HPLC or GC, and your sample prep items. So your syringe filters, your syringes, your SP cartridges that go on a vacuum manifold or a positive pressure manifold, as well as our micro SPE plates that we have. We have Craig in the back, who will also be speaking today. Craig is part of our Center of Excellence team. He is a metabolomics guru. So that's why I brought him in to talk about the application-specific metabolomics. Your account manager in terms of anything else outside of chromatography consumables is Drew Smiley. He will be with you guys for any instrumentation, anything you need outside of me or Craig, or even with Craig and myself as well. So Drew is kind of your main person of contact. So Drew is here today. You've got some other stuff, somebody else today as well, right? Matt, is Matt here doing training? Okay. So we cover the entire University of Florida campus. I want to thank you guys for all coming to the talk today. If you haven't already used the QR code to kind of log in, it makes it a little bit easier than trying to decipher some handwriting. I'm really bad at that. So we have a metabolomics heavy conversation today that we wanted to discuss. I'm going to go into the column chemistries that are best used for metabolomics. Craig, like I said, is going to go into an application-specific metabolomics application. And he'll go over the chemistries and what chemistries were used and the instrumentation that was used for it as well. Do you want to wait a little longer? Do we just want to go with it? Sure.
Go ahead. Yeah.
We got a couple more logging in now.
Yeah.
Perfect. This was working. Technology is great until you need it. All right. So HPLC columns for metabolomic analysis. First, I guess the best thing is to actually define the word metabolomics, right? What is metabolomics? So metabolomics is your large-scale study of small molecules. They are commonly known as metabolites, obviously. And then within the cells and biofluids, tissues, and organisms, these collectively small molecules are their interactions within the biological system. That's actually called the metabolome. So that's this part here is the actual metabolomes. And then you go from the metabolomes into the more specific metabolites. The metabolites are their concentrations, unlike other omics measures, directly reflect the underlying biochemical activities in the state of the cells or the tissues. Metabolomics and the endpoint of the omics cascade and provide unique insight to biology from a phenotype perspective. So it kind of gets super specific in terms of what it actually is. Metabolomics as an application. Metabolomics is an extremely broad topic to begin with. It covers a wide variety of applications. As you can see here, this is just the human and health side of things. So you can cover cardiovascular, personalized medicine, aging, biomarker discovery. Then you can also do it in the industrial research, which is your drug development, your food research, your plant research. So it's a common theme of research, but it is a very broad topic in terms of what can qualify as a metabolomics application. With such a broad topic comes its issues or its complications or challenges. To name a few, you have the complexity of the biological sample. Is it blood, urine, tissue? what kind of tissue? Is it coming from the brain? Is it coming from a stem cell tissue? We have the wide range of metabolite concentrations. That's going to affect what kind of instrumentation you're using or the type of column you're using as well. You have isobaric and isomeric species. Usually those will elute the same time in a column. So picking your proper column chemistry is going to help with that particular challenge. And then you have not all features are equal metabolites, which that's kind of like saying everybody in this room is the same as well. So with that, we have 3 different types of chromatographic separation that's usually pretty common in the metabolomics world. You have LC, then you have just regular chromatography, and then you have your IC chromatography. When we're looking at metabolites, we have a broad psychochemical properties that also range in polarities, ionization, which all depends on how you're going to separate it chromatographically. You don't want to use a C18 for a superpolar compound. Probably not going to work the best. So based on that, you're also going to look at your MS ionization as well, whether your mass spec is going to be in positive mode or negative mode. So we have our 4 main types of chromatographic separation. We have a reverse phase column, which is the most normal phase column that customers are going to use. I use the word normal phase very loosely in this situation. It's the most commonly used separation is a reverse phase. There is a normal phase column as well, but not for this. We have our Helix or a modified Helix. We have our ion pairing reagent with reverse phase. And then we also have our ion chromatography. The second 2 are going to be a little bit more different. And the reason I say that is because ion pairing reagents are not always the greatest on mass specs. So if you have a mass spec, probably don't want to go with that particular separation. Then you have ion chromatograph, which we have an instrument specific to IC or ion chromatography. We have 3 main column families at Thermo. We have our hypersil gold, which is, we call it our general purpose. It's kind of our one-all be-all, but we also have our hypersil gold specific columns as well. So even though it's generally a good C18 column, we also have our acclaim columns and our AccuCore columns that also come in a C18 phase. Our acclaim column is going to be a fully porous column or particle column with better resolution. Our AccuCore column is a solid core particle, which is actually going to allow for a higher throughput and also pretty high resolution and separation as well. Based on your application, depends on which kind of column family you're going to go with. These are all the different kinds of columns we have within those families. Personally, I don't know every single one of these. I wouldn't expect you guys to either. We have multiple tools that help kind of narrow down what kind of column you want to look for or the kind of column you want to use per your application. The theme of this is what kind of application are you working on? What kind of application are you doing? I don't want to give you guys a column that's not going to work for your application. So based on your application, we can stick with the AccuCore columns, the AccuCore XL, or the Vanquish. We have the hypersil gold family, which is a general purpose reverse phase column, an ion exchange column, or a Helix column. We have our acclaim family, which is going to have something a little different. It's going to have our mixed mode columns as well as application specific columns. So this is another thing where if you're looking for something very specific for your application, and that is a champion column for your application, try the application specific ones first. But we still have to consider what we're actually looking for. So in order to pick which family, then pick which column, some things to consider are the dimensions you're looking for. Are you looking for a 1.0 millimeter column, a 2.1 column? We go all the way up to a 4.6 in regular analytical columns. Our prep columns is a completely different story. They go to the size of a dumbbell and are very heavy. We have different lengths. We go from 10 millimeter, 30 millimeter, 50, 100, 150, and 250. Those are going to be our 6 most common lengths that we have. But a lot of it's going to depend on your flow rate of your LC and your pressure that your LC can withstand. You don't particularly need a column that goes up to 15,000 PSI if your HPLC doesn't even go that high. It's great if your column can outdo your HPLC, but that doesn't require you to have an HPLC like that. We also have our capillary and nanoflow. Jin's lab has how many nanoflow systems? 2? 5 nanosystems. So he's not going to put an analytical column on something that needs low flow rates. So that's also going to narrow down what kind of column chemistries you're going to look at as well. So we're going to focus on 2 main ones that I discussed earlier. We have our reverse phase and our Helix columns. The ion pairing, if you have a mass spec, again, is probably not the best option. And then you have the IC, which if you're using an HPLC, you're not using an IC in this particular application. Not to say you can't, but we're not going to talk about that today. Our reverse phase columns, again, you're going to see this hypersil gold column very frequently. So one of the columns that we recommend right off the bat when we hear metabolomics for reverse phase is our hypersil gold column. It sits at a 2.1 and a 150. That gives you enough column to separate the compound you're looking at. And it has a large enough internal diameter, which is going to allow you to put a flow rate that gets your stuff through your column at a decent rate. So it's, for this particular one, is a 15-minute method. So you're going to get good separation on a 15-minute method. And this is just an example HPLC flow rate that we're looking at. So you can see that the flow rate is sitting at 0.3 mils per minute. And then there's a varying concentration for your organic and your aqueous solutions. We have our Helix column. You can do it in positive mode or negative mode mass spec, depending again on your particular compounds. The Helix that we're looking at is an AccuCore. If you remember from the previous slide, we had the hypersil gold and the AccuCore columns for Helix. We recommend the 150 Helix or the 150 amide Helix. Your Helix columns are going to be more for your very polar compounds. And again, we're sticking to that 2.1 millimeter. So that's a decent internal diameter and usually a pretty common one you're going to see. The big difference between these isn't just the 150 and the 100, but it's the 1.9 and the 2.6 particle size. Our AccuCore with a solid core particle is going to essentially be a little bit bigger anyway, because that actual center core is about the same size as the actual pore in a fully porous hypersil gold. So to add that extra width to the column or to the particle, that's where you're going to get that 2.6. So it's a solid core, but you still have to get your funnels into the core. So if we go back real quick, these are the little funnels that I'm talking about. So you see that you have in the AccuCore, they're purple in this situation, going into that yellow ball. That yellow ball is your solid core. So that's about the same size as your hypersil gold particle to begin with, which has just the cones going into the center. So what that fully porous core is actually going to do is allow the compounds to flow through the particle. We're also sticking to the 15-minute cycle, as well as increasing actually the flow rate to a 0.5 millimeter per minute. So again, these are just kind of starting positions for you guys on a application. This is generic. This is not super specific based on your application. Some of these things might change based on the hydrophilic side of your compounds, or whether or not they are going to be more basic or acidic, and if they're going to be positive mode or negative mode. Sometimes you might need to extend your actual method depending on your mass spec polarity switching. If it doesn't switch as fast as you'd like it to, then you might have to extend your flow rates or your time in order to actually get the mass spec to switch in the time frame that it needs to. Even though those are our recommended reverse phase columns, we have plenty more to look at as well. We have our C18, we have our AQ. So that's going to be something that's run in a very aqueous mobile phase. We have our polar premium. We have our reverse phase mass spec. So the reverse phase, the RPMS, those are going to be the columns that you're using on, let's say, clinical high throughput mass spec analysis. So something that's under 7 minutes with a very high flow rate where you're just trying to blow things through just to get the method done. Because it can withstand that pressure background with good separation still. We have our C8, phenylhexyl, phenyl X, biphenyl. We do only have one type of biphenyl in our families, and that is our AccuCore. We have our PFP, our C30, and again, we see that Vanquish C18 plus. Yeah. Then we have our Hypersil Gold. One thing to point out is the particle size of our AccuCore. Like I said before, it's the 2.6. It does not vary, except for the C18 plus. If you look at the Hypersil Gold and the Acclaim, that's where you're going to get more particle differences between the actual columns. That's also going to give you your largest variation between the columns. If you'd like to try different particle sizes, there's the 1.9, the 3, and the 5 in the Hypersil Gold. We up it a little bit in the Acclaim with the 2.2 particle size, which also is a 3 and a 5. The pore sizes are a little different between the two, even though they're still essentially the same type of column with the fully porous particles. They are going to differentiate in the pore sizes as well as the surface area. So there's a couple things to look at when we are comparing columns that you might already be using somewhere along the lines. So we have Phenomenex, we have ResTech, we have Waters, we have Agilent, we have Shimatsu, and then we have Thermo. Everyone's got a C18 column. So what's going to make you choose a Thermo C18 column over an Agilent C18 column? It's going to be application specific, right? I keep going back to that. I particularly like to sell my columns to customers in the application space rather than just a general purpose, hey, try this column. My goal is to make sure that you guys actually like your column and you're going to keep using your column and you're happy with your column. So it might take a little bit of extra time to determine what kind of column you're looking for, but as long as it does what you need it to do, then let the time take as long as it needs to take, right? Nothing worse than spending money on a column to turn around and it be the wrong column or not work the way you want it to. So we go into our LC selections based on the actual metabolite. So based on the polar metabolite, medium polar or non-polar, you have your actual LC modes that you're looking at. It seems the most common LC mode is your reverse phase. But again, depends on the type of metabolite you're looking for. If you're looking at a polar metabolite, you're going to go Helix because that's the type of column that's going to be needed for that kind of compound. If you use a reverse phase, it's not going to stick. It's not going to stick at all. I don't know if anyone's ever tried to analyze glyphosate on a reverse phase column. Glyphosate is a tiny particle, very tiny compound, and it's also a very polar compound. So when you try to run it on a regular C18 or a biphenyl, it just is gone. It just blows right off. There's no way to keep it on. If you use a Helix, you have a little bit more retention for it. So a Helix stationary phases. So we went over a reverse phase, which is our C18, biphenyl, phenyl, just to name kind of the common ones. Now we go into our Helix. So we have Helix, and then we have a silica. There we go. So each one of our families has a Helix column. It depends on what you're using it for again. Remember, we talked about the AccuCore having its own like specialty columns, as well as the acclaim having its specialty columns. The synchronous silica and the synchronous helic, they're there. We call those our legacy columns. They are still an option, but are slowly being faded out. So that's why we kind of talk about our three main families as the hypersil gold, the AccuCore, and the acclaim. So again, based on your need for a polar metabolite analysis, these are the different kinds of phases that are going to be with those columns. The phases matter. That's the whole purpose of this particular presentation, is the phases and the column chemistry. So if you need something that's going to work a little bit better with an amide, probably the AccuCore amide helic column will be one that would be recommended. Then we would just have to go into whether or not you want it's going to be a 2.6, but then we can determine what kind of size you need based on the length or the internal diameter. This is just another example of basically that particular table in a form that looks a little bit more visually pleasing, right? We have our high bases, which are going to be our helic, which are also our superpolar compounds. Then we have our neutral compounds, which are also in the Helix Phases. And then our hypersil gold acidic acids, or acidic analytes. So this will be the more visually pleasing version, like I said, of that particular table of the Helix side of things. So here it doesn't really talk about the acidity or any sort of pH of these particular compounds, which again, if your pHs are going to be at a varying pH, this will also help you determine which kind of column is going to be your best one. We have our C30 column, which is a higher shape selectivity. So basically what that means is you have more theoretical plates when you're looking at the actual column peak. So we have our Acclaim 120 C18 here. And as you can see, the 30 kind of separates a lot of that extra stuff at the bottom into actual peaks. So there's a little bit more resolution on those. It might not be the greatest because you don't necessarily see the baseline resolution, but you see that there's additional peaks. So what once was a shoulder is now an actual separated peak. Same method, same LC method, same parameters on the LC. The only thing that changed was the column. It's one of those things where it shows how much a column chemistry can change your actual column separation. We do have a paper, just for lipidomics side of things, we do have a paper for the C30 column. And it also shows the number of identified lipids. So again, that goes into the actual separation of those shoulders and those compounds that are separated. When you use the C30 column, you have 1,300 different identifications versus the C18A, B, and C column. They're all either ours or competitor columns in a fashion that are under anywhere between 900 and 800. So to get more compounds, they're using the C30 because of that particular resolution and that separation. Application specific. We're going back to those Acclaim Trinity columns. Depending on what you're looking for, these are going to be your powerful columns for your application specific things. So these are the column chemistry inside of these are made specifically for certain compounds. So they're going to have a reaction based on a certain family of compounds. This is going to be an example of the quantitation of amino acids with the Trinity P1. So you can see all of these different compounds have been separated. This was on a triple quad Quantus in an 18-minute method. So using the Trinity P1, it allows you to actually see the amino acids, which if anyone's tried to analyze amino acids on other columns, it's not always the easiest. So they're showing here the fact that it can be done with 42 different amino acids. They are all individually identified and for the most part separated based on M or M transitions. So going back to the LC requirements for any sort of application that you're looking for. Main questions to ask are, stationary phase are you looking for? How do you determine that stationary phase? Are you looking for selectivity? Retention? Everyone's looking for reproducibility, right? You don't want to have one column and then one day it works and the next lot of columns doesn't work. Everybody wants reproducibility from day to day, lot to lot. Stability. Stability is another one that's super important when it comes to large applications that are going to be run over a period of time. Column dimensions, again, that goes back to whether or not you're looking for an analytical column or a low flow column. If you're looking at low flow columns, we have microflow. Are you looking for nanoflow, capillary flow? I do have an example of our micropack column in the back. That is a microflow column. It is from Pharmafluidics, but it is now going, we have a Gen 1 that's released now, and then our Gen 2s and 3s will be released later this year, March. So when you're looking for columns and you're looking at applications, don't be afraid to ask yourself those questions. Don't be afraid to ask me those questions and to use resources that are available. We have an LC column tool that you can look at a method. You can even look at competitor columns and say, I've got this particular competitor. Let's see what matches with that column for thermo. I want to give thermo a try. Let's see what we have. So there are many tools that allow you guys to have an idea of the kind of columns you're looking for. The other 3 great resources that you have are sitting in this room today. You have myself, Craig, Drew. So when you have questions like these, don't be afraid to ask the question, because that is why we're here. We're here to be your resource to help your applications succeed and to help you guys succeed in your applications. Todd Stoner is the only person that I did not mention today earlier. Todd is your technical support specialist for this area. You may have ran into him over the past couple of years throughout. There's been a reorganization and now Todd is a technical support specialist. He is here to support the team as a technical resource in the mass spec in the LC world. He is phenomenal. So those are the 4 main people that you guys can contact in terms of metabolomics, HPLC, GC, GCMS, LCMS, any sort of application you're looking for, any sort of technical or regular questions you have. Do you guys have any questions? Thank you. Absolutely. I can definitely send that.